Did you know that the ice that we use to cool our drinks in the summer is not the only type of ice that exists in the universe, and it's not even the most common one? Scientists have discovered that water can freeze in different ways, depending on the temperature and amount of pressure applied during the freezing process. As it turns out, the ice that exists in space, called amorphous ice, is likely the most common way for water to exist in the universe. Scientists have found a way to create this glassy space ice on Earth, and have used it to explore what life looks like inside living cells. Before we get to that, let's look at why the way water freezes is important in microscopy. To look at life at the smallest scale, first you need to immobilize it. Typically, this is done by fixing and dehydrating the samples. However, these processes distort the natural state of life and thereby impact how structures appear under a microscope. This prompted scientists to look for different ways to preserve their samples. It took time and collaboration to find a solution to this problem, and one of the key researchers involved in this quest was a Swiss-French scientist who worked at EMBL, biophysicist and Nobel laureate Jacques Dubochet. One of the issues was that biological samples are mostly made of water, and when you freeze water, its molecules try to rearrange themselves into crystals, which take more space than liquid water. These crystals damage the samples by disrupting the cells. What the scientists discovered was that if you freeze your samples very fast, crystals don't have time to form. You are creating amorphous ice on Earth, which is crystal-free. Freezing cells this way allows immobilizing them without altering their structure. The method they used for the tiniest samples, smaller than the width of spider silk, is called plunge freezing. It's called this because you drop the sample into a liquid at -200 degrees Celsius, which cools it down very quickly. Another technique to produce this amorphous ice, used for larger samples, is high-pressure freezing. It uses both a high pressure of 2,000 times the one of the atmosphere at sea level and a very low temperature of -200 degrees Celsius to very quickly lock biological samples in space and time. Imagine a tiny pebble, squished at the bottom of a mountain higher than Everest and almost 3 times colder than Antarctica in winter. Once we have a quickly frozen sample, we can remove the high pressure and keep the low temperature, and there we have a sample ready to be observed with an electron microscope. The combination of high-pressure freezing and electron microscopy is very powerful and produces incredibly detailed pictures of tiny structures at the molecular and atomic level, allowing scientists to observe life as it is. This is important because only by seeing life as it is can we understand how it works and truly enjoy it.